There are three types of RNA that are going to be used in the protein synthesis process. There is messenger RNA, otherwise known as mRNA, and this is basically a linear copy of the DNA code. And it includes start sequences and stop sequences for the code. And remember that one code or one piece of this would represent one gene, therefore one protein that is being copied. The second type is ribosomal RNA, or rRNA. This is made up of large and small subunits, and that we call them, and these are found in the nucleolus. So the ribosomal RNA is kind of, there's already some present in the nucleus, and there may be more made for the process. Transfer RNA is the third type. Transfer RNA transfers amino acids to the process of protein synthesis from the cytoplasm to the rough ER. Transfer RNA looks like this. That it is RNA, and it looks kind of like a little club here. And it has a particular portion down here called the anticodon that will match with the messenger RNA. And we'll talk more about how it does that in a little while. But this anticodon is associated with this part up here, which is going to pick up a specific amino acid. So anticodon matches the codon, matches a specific amino acid, and in this way, specific things can be put into place. And we'll talk more about that in a bit, okay? Now let's just talk briefly about the process of making RNA. I don't want you to know this whole process or anything, but I just kind of tell you sort of an overview. Basically, it's similar to replication in that we're going to use base pairing to copy the DNA code. But again, this is only one gene, so there is a part that is going to be recognized as the kind of the beginning. We have enzymes, of course, that are part of this, promoters that are starting us off. Then we start copying the RNA with base pairing, everything the same except for uracil, like we talked about in the last little snippet of lecture. And then it's going to move along the DNA, copying just one side of the DNA, just like in replication. But in this case, the DNA is going to close up behind it, and the piece of RNA does not stay attached. It comes off, or so it'll be hanging off of the DNA as the process continues, all right? And then when we get all done, we're going to have that piece of RNA is going to be loose, and the DNA place that was copied will be completely closed back up and unaffected, all right? So that's kind of an overview of how it would happen. This is like, whoa, I got two of them there. This is a sort of another depiction showing you the enzyme RNA polymerase and how it's adding the RNA nucleotides base pairing with the DNA. So this is DNA down here, and this is the RNA. And the main difference between this and replication is two main differences. One is only one half, and the other one is it's hanging off and the DNA is closing back up behind. Okay? Now another thing is, let's see, that's not what I wanted to show. All right, so another thing is that when a gene is copied, a lot of times most of the time in a gene or the copying sequence, there will be parts that I will not use. In other words, in terms of making the protein in question, there will be parts of the DNA that are, and essentially in this case, junk. They're not really junk, but they could be used perhaps by another gene or in another cell in another way or something like that. But anyway, generally speaking, we have the things called exons and introns. Exon, intron, exon. And what has to happen is, so I copy that whole thing in that sequence that I was just talking about, and then I have to cut out the introns, which are basically not used. So the exons are what's used, and the introns are going to be cut out. So when I finish my RNA, that is, when I finally finish making it, so I'm going to transcribe the RNA. That's a process. It's replication of the RNA. So they give it another name called transcription. And then they call it processing, where they're cutting out the introns. and they're going to then have the codon or the messenger RNA that we're actually going to use is going to include the exon 1, which is green, the exon 2, which is yellow, and the exon 3, which is purple. It's going to include all those in one long line, which is my messenger RNA piece that I'm going to use for the process.